Hello students, and welcome back to the Community Nutrition series of videos. In today's video we're going to talk about microbiota and community nutrition and genetics and community nutrition. So expect to learn how the microorganisms we have in our body will affect our health, and also how our genetic will influence our nutrient selection, and vice versa, how our nutrients can affect our genetics. So let's begin. Hi, I am Tommy. I'm a human nutrition student and researcher at the university. What I do in this type of videos is to synthesize what I learned at the university during the classes, so then I can explain and teach you the best and most valuable information you need to know. So let's begin by talking about the microbiota. The microbiota. Definition. These are microorganisms of a certain place, and we do not call them flora. So microbiota, who are they? What microorganisms? Microbiome are the genes of those microorganisms in the microbiota. What they do? Characteristics. Well, microorganisms are in 1:1 ratio to human cells. Very interesting. And there is approximately 1 kilogram of microbiota mainly in our intestine. We cannot live without microbiota. Very important. And this microbiota is constantly changing. The functions of the microbiota are mainly three. Metabolic, it helps us digest compounds. Immune, it defends us from diseases, also chronic diseases. And structural, it reinforces our tight junctions to prevent the leaky gut syndrome. Very important. So the microbiota mainly defends us and also protects us. Main types of microbiota, we have the Bacteroides, Bifidobacteria and Lactobacillus. There are also other types, but these are the main ones. They can be autochthonous, which are resident in that area, or alloctonous, which are transient. We gain and change the microbiota during our lifetime. And there is a variation in the birth type as well. A natural birth always gives a better microbiota to the child. When taking a look at those microorganisms we have, we call eubiosis when there is a balance between them, and it will lead to health. And a dysbiosis when there is an imbalance, and this is correlated with disease. Dysbiosis disorders will create immune, metabolic, neurological, and digestive problems. Also cardiovascular, respiratory, and renal disorders in female reproductive system because of those dysbiosis. The human microbiota. We have microbiota in the skin as a protective function, and it is used to teach the immune system. There are different types of microorganisms in different parts of our body. It's not the same our feet than our abdomen or our chest or our back or our butt. And a dysbiosis in this skin microbiota will lead to acne, psoriasis, atopic dermatitis, and rosacea. Then there is a vaginal microbiota. This has a very low diversity. It is mainly lactobacillus. And the dysbiosis in this part of the body will create miscarriages, premature births, infertility, or inflammatory diseases. This is called bacterial vaginosis, and it is mainly derived from what we eat. And there is this term, estreboloma, which is the capacity of a microorganism to create estrogens, to regulate estrogen levels. And then the oral microbiota. There is a high diversity here, much higher compared to the vaginal microbiota. This microbiota is constantly changing during the day as well. It's an indicator for future diseases in the rest of the body. Some bacteria in the mouth is correlated with heart diseases. For example, if we have cavities, the probability of heart disease is higher. The dysbiosis is due to antibiotic drugs, tobacco, and also diet. And research has shown that kisses can improve the microbiota and even prevent dental cavities. Yes, because there are two main species of bacteria, Streptococcus dentisani and Streptococcus downi, that do protect teeth from decay. And the dysbiosis is mainly done because of Streptococcus mutants that will create acid and indeed create cavities. Moving on, we have the gut microbiota. This is very high diversity. More than 1,000 species are present in our gut. But this is decreasing, this number, because of our Western lifestyle. Mainly we have Bacteroidetes. These are Bacteroides and Prevotella, and Firmicutes, Lactobacillus and Clostridium. A dysbiosis in these microorganisms will lead to leaky gut syndrome. And the leaky gut is associated with inflammatory bowel disease, ulcerative colitis, Crohn's disease, and Helicobacter pylori. These microbiota are, of course, different microorganisms, and there are different roles depending on the microorganism. Some of them are for the fatty acid creation, such as acetate, propionate, and butyrate. These are very short-chain fatty acids. These are very good for our health. Some of those microorganisms will create vitamins and enzymes, and some of those will create cytokines. Again, our gut microbiota is there to protect us and help us digest food. And then we have the microbiota. Microbiota is the study of fungi. Some of those fungi species can regulate the gene expression in the hippocampus. And this is very interesting because Candida cephei, the cephei is the fermented milk, can also improve encephalomyelitis in mice. So as you can see, some microorganisms have a pretty powerful capacity of improving our health. And then we have microbiota and diseases, of course, due to dysbiosis. We can have immunological diseases such as celiac disease and leaky gut, metabolic diseases, obesity and diabetes, very common nowadays. Neurological diseases such as Parkinson's and Alzheimer, they are associated with dysbiosis in the gut, the second brain. Mental diseases, anxiety and depression, autism and ALS, amyotrophic lateral sclerosis, are also associated with a dysbiosis of microbiota. And of course, digestive diseases, hepatitis, SIBO, which is small intestine, bacterial overgrowth, vaginal diseases, and sarcopenia. So what are the recommendations then? Do not abuse antibiotics, tobacco, or alcohol. Exercise regularly, reduce stress levels, get a good night of sleep. Sleep hygiene is very important. Probiotic ingestion and prebiotic ingestion to have that symbiotic effect. Probiotics, again, are microorganisms. Prebiotics are ingredients for those microorganisms to grow. Fiber. And in case nothing works, there is also a bacteriophagy therapy which has been studied by the Soviets to kill the bacteria instead of using antibiotics. Again, this is to solve a dysbiosis. And now it's time to take a look at nutrigenetics, nutrigenomics, and community nutrition. This is how the genes can affect our health, and how what we eat can affect our genes. Yes, because nutrigenetics, gene to nutrient. It's a science that analyzes how the body responds to nutrients based on genetics. And it establishes a nutritional recommendation based on genetic predisposition. On the other hand, nutrigenomics, nutrient to gene, studies how nutrients affect gene expression. So with nutrigenetics, we can find the perfect diet for an individual because of the predisposition. And with nutrigenomics, we can take a look at how the food will affect our gene expression. And how can we test this? With epigenomics. This is the science that improves genetic characteristics through artificial selection. What are the mechanisms? DNA methylation, modification of histones, and regulation by non-coding RNAs. A DNA methylation is a cytosine, the nucleotide, cytosine methylation at the side of a guanine. So when we have a cytosine near a guanine, we methylate the cytosine and there is an inhibition of the gene expression. Modification of histones, packaging of DNA so that it cannot be expressed. Basically, we modify histones to prevent the spread of the DNA chain so the transcription will not occur. And regulation by non-coding RNAs, these are 20 to 22 nucleotide strands that silence the gene expression by sticking to the mRNA to form a double-stranded RNA. So this is basically a second RNA that will bind to the real RNA, impeding that gene expression. And the diet influences the DNA methylation pattern, for example. As you can see here, from diet to epigenome is nutrigenomics, and from epigenome to diet is nutrigenetics. Differential gene expression, different metabotypes, and the microbiota will also affect the epigenome. The studies that have been done in genetics are mainly conducted in white race, so they have little applicability to other groups, unfortunately. But what we discovered is that there are mutations. It can be a genomic mutation, which involves the DNA, epigenomics, DNA, transcriptomics, RNA, proteomics, protein, and metabolomics, metabolites. These are all indeed going to change the phenotype, the gene expression. So let's take a look at some examples. Genetic variants in apoprotein E predispose to Alzheimer's disease. The MC4R misregulation can be reversed by a high-protein diet. The GSTP1 transferase failure increases predisposition to colon cancer. The PPAR mutation increases blood lipids, and polyunsaturated fatty acids in the diet help correct the dyslipidemia. The FUT2 mutation is associated with lower vitamin B12, and the VDBP mutation is associated with lower vitamin D. And affecting alcohol metabolism, an alcohol dehydrogenase mutation is also associated with higher efficiency of the enzyme, will create a greater hangover because the alcohol is metabolized to aldehyde. And the aldehyde dehydrogenase mutation is always associated with a lower efficiency of the enzyme, is also associated with a greater hangover because the aldehyde is not metabolized and it accumulates. So the bioaccumulation of aldehyde equals hangover symptoms. And now let's take a look at chrononutrition. This is very interesting. It's the understanding that meal timing can affect our health. And how? It's based on the relation between nutrition and the circadian rhythm. The goal here is to eat according to the circadian rhythm. So when starting a day, we shouldn't really eat as soon as we wake up. It's not physiological. Same thing to eat late at night. We shouldn't really eat very late at night. It's much better to follow a diet during the sun hours. And I know in certain countries that's not possible, unfortunately. So the goal is to eat according to the circadian rhythm. Why? Because the chrono disruption or the circadian rhythm interruption can lead to diseases, even chronic diseases. So it's very important to try to follow a diet that matches with the diurnal hours and that follows your circadian rhythm. Genetic tests, then. These are used for diagnostic, presymptomatic, carrier detection, prenatal, predisposition, pharmacogenetics, nutrigenomic, behavioral, family, relationship, and ancestry. These are all the types of tests that we can find out there. These are genetic tests, but they may be unreliable and they can give false positives. Also, these genetic tests give data to companies that can be used to control and charge more for the same services. Imagine, for example, that you have a genetic predisposition of Alzheimer's disease. Well, chances are a clinic that treats Alzheimer, if they know that you have this predisposition, they are going to charge more without you knowing because they know you have that predisposition. And it's very important that the patient's integrity must be adequately protected. So it's very important to know this, to not fall for these genetic tests tricks. Okay, this is it for this video. I hope you understood everything I said. If you didn't, there is a comment section down below. Don't forget to like, subscribe, and turn on the notifications so you get notified every time I upload a new video. If you enjoyed this minute with me, don't hesitate to share it with your friends to spread this knowledge to them as well. Thank you so much for watching, and I will see you in the next one, in which we are going to talk about the eating habits of the population, the labeling on the food, as well as the nutritional policies. So stay tuned for more knowledge on nutrition. I'll see you next time. Ciao!